Dear students, Assalamu alaikum. We will continue with the acid-base regulation. The objectives of today's lecture are to learn about secretion of hydrogen ions, reabsorption of filtered bicarbonate ions, production of new bicarbonate ions to regulate extracellular fluid hydrogen ion concentration. In the last lecture, we learned some general points about the renal control of acid-base balance. Continuing with that, a very important point to note at this juncture is that the kidneys regulate extracellular fluid hydrogen ion concentration through three fundamental mechanisms. Number one, secretion of hydrogen ions. Number two, reabsorption of filtered bicarbonate ions. And number three, production of new bicarbonate ions. Hydrogen ion secretion and bicarbonate ion reabsorption occur in all parts of the tubules except descending and ascending limbs of the loop of Henle. A very important fact to memorize is that for each bicarbonate reabsorbed, a hydrogen ion must be secreted. Now let's study the first of these fundamental mechanisms, which is secretion of hydrogen ions. As mentioned earlier, the mechanism of bicarbonate reabsorption also involves tubular secretion of hydrogen ions, but different tubular segments perform this task, that is, secretion of hydrogen ions, differently. To study secretion of hydrogen ions, we will divide the renal tubule into early and late segments. The secretion of hydrogen ions in early tubular segments is by secondary active transport, and the secretion of hydrogen ions in the distal tubular segments is by primary active transport. Secretion of hydrogen ions in early tubular segments. The epithelial cells of early tubular segments secrete hydrogen ions into the tubular fluid by secondary active sodium hydrogen counter transport. This secondary active secretion of hydrogen ions is coupled with the transport of sodium ions into the cell at the luminal membrane by the sodium hydrogen exchanger protein. The energy for hydrogen ion secretion against a concentration gradient is derived from the sodium gradient. The sodium gradient is established by the sodium-potassium ATPase pump in the basolateral membrane. The figure shows the cellular mechanisms for active secretion of hydrogen ions into the renal tubule and sodium reabsorption in exchange for hydrogen ion secreted. This pattern of hydrogen ion secretion occurs in the proximal tubule, the thick ascending segment of the loop of Henle, and the early distal tubule. Hydrogen ion secretion also achieves reabsorption. Carbon dioxide combines with water to form carbonic acid. Carbonic acid dissociates into hydrogen ions and bicarbonate ions. The hydrogen ions is secreted from the cell into the tubular lumen by sodium hydrogen counter transport. Bicarbonate ions move downhill across the basolateral membrane into renal interstitial fluid and the peritubular capillary blood. The net result is that for every hydrogen ion secreted into the tubular lumen, a bicarbonate ion enters the blood. Coming to the secretion of hydrogen ions in distal tubular segments, the characteristics of hydrogen ion secretion in these segments are different. The tubular epithelium secretes hydrogen ions by primary active transport. Primary active secretion of hydrogen ions occurs in type A intercalated cells of the late distal tubule and in the collecting tubules. Hydrogen ion is secreted into the tubule by number 1 hydrogen ATPase and number 2 hydrogen potassium ATPase. For each hydrogen ion secreted, a bicarbonate ion is reabsorbed as observed earlier. This figure shows the active secretion of hydrogen ions through the luminal membrane of the type A intercalated cells of the late distal and collecting tubules. Type A cells contain hydrogen adenosine triphosphatase and hydrogen potassium ATPase in the luminal membrane and secrete hydrogen ions while reabsorbing bicarbonate ions and potassium ions in acidosis. Note that one bicarbonate ion is absorbed for each hydrogen ion secreted and one chloride ion is passively secreted along with the hydrogen ion. Hydrogen ion secretion in these segments is important in forming maximally acidic urine. Hydrogen ion concentration can be increased 900-fold in the collecting tubules. Reabsorption of bicarbonate ions. You may recall that this is the second fundamental mechanism used by the kidneys to regulate extracellular fluid hydrogen ion concentration. A very important point to keep in mind is that the bicarbonate ions cannot enter the luminal membranes of the renal tubular cells. Therefore, filtered bicarbonate cannot be directly reabsorbed. I repeat, cannot be directly reabsorbed. Bicarbonate is reabsorbed by a special process in which it first combines with hydrogen ions to form carbonic acid. Carbonic acid dissociates into carbon dioxide and water, and carbon dioxide can easily move across the luminal membrane into the tubular cell. Inside the tubular cells, carbon dioxide combines with water to form carbonic acid. Carbonic acid dissociates into hydrogen ions and bicarbonate ions. And bicarbonate ions moves downhill across the basolateral membrane into the renal interstitial fluid and then into the peritubular capillary blood. The figure shows the cellular mechanisms for tubular reabsorption of bicarbonate ions by combination with hydrogen ions to form carbonic acid, which dissociates to form carbon dioxide and water, and carbon dioxide later diffuses into the luminal membrane of the renal tubular cell. Once inside the renal tubular cell, the transport of bicarbonate ions across the basolateral membrane is facilitated by two mechanisms. Number one, sodium bicarbonate co-transport, especially in the proximal tubules, and chloride bicarbonate exchange, which occurs in the late segments of the proximal tubule, thick ascending loop of Henle, and the collecting tubules and ducts. The net effect of these reactions is reabsorption of bicarbonate ions from the tubules. Although do remember that the bicarbonate ions that actually enters the extracellular fluid is not the same as that filtered into the tubules. Titration of bicarbonate ions against hydrogen ions. Normally, the quantities of bicarbonate ions and hydrogen ions entering the tubules are almost equal. They combine with each other to form carbon dioxide and water. So, it is said that bicarbonate ions and the hydrogen ions titrate each other in the tubules. However, in metabolic alkalosis, there is an excess of bicarbonate ions over the hydrogen ions in the urine. And this excess bicarbonate ions cannot be reabsorbed. Therefore, the excess bicarbonate ions are left in the tubules and excreted into urine, which helps correct the metabolic alkalosis. In acidosis, there is excess of hydrogen ions over bicarbonate ions, causing complete reabsorption of bicarbonate ions. The excess hydrogen ions pass into the urine in combination with the urinary buffers, phosphate and ammonia, and is eventually excreted as salts. The third fundamental mechanism used by the kidneys to regulate extracellular fluid hydrogen ion concentration is the production of new bicarbonate ions. So how this occurs, this production of new bicarbonate ions? The combination of excess hydrogen ions with phosphate and ammonia buffers in the renal tubule produces or generates the new bicarbonate ions, as we shall study a little while later. Combination of excess hydrogen ions with phosphate and ammonia buffers. When hydrogen ion is secreted in excess of the bicarbonate ions, only a small part of the excess hydrogen ions can be excreted in the ionic form in the urine. When there is excess hydrogen ions in the tubular fluid, it combines with buffers other than bicarbonate ions, leading to generation of new bicarbonate ions. And the important buffers for this purpose, for the generation of new bicarbonate ions, are phosphate buffer and ammonia buffer. Phosphate buffer system. As mentioned earlier, the phosphate buffer system is composed of hydrogen phosphate and dihydrogen phosphate. Once all bicarbonate has been reabsorbed and is no longer available to combine with hydrogen ions, any excess hydrogen and ion can combine with the hydrogen phosphate ions. After the hydrogen ions combine with the hydrogen phosphate ions to form dihydrogen phosphate, it can be excreted as a sodium salt, carrying with it the excess hydrogen ions. There is, however, one important difference in this manner of hydrogen ion excretion from that we discussed previously, which is that in this case, newly generated bicarbonate ion in the tubular cell entering the peritubular blood represents a net gain of bicarbonate ions by the blood, rather than merely a replacement of filtered bicarbonate ions. Therefore, whenever a hydrogen ion is secreted into the tubular lumen, combines with a buffer other than bicarbonate ion, The net effect is addition of a new bicarbonate ion to the blood. This diagram shows the buffering of excess secreted hydrogen ions by the filtered phosphate. Note that a new bicarbonate is returned to the blood for each sodium hydrogen phosphate that reacts with a secreted hydrogen ion. Under normal conditions, most of the filtered phosphate is reabsorbed. Therefore, a greater proportion of excess hydrogen ion in the tubular fluid is, like in acidosis, occurs through the ammonia buffer system. Ammonia buffer is composed of ammonia and the ammonium ions. Ammonium ion is synthesized from glutamine coming from metabolism of amino acids in the renal tubular cells. In the proximal tubules, thick ascending limb of loop of Henle and distal tubules, the glutamine is transported into the tubular epithelial cell, and once inside this cell, each molecule of glutamine is metabolized to form 2 ammonium ions and 2 bicarbonate ions. The ammonium ion is secreted into the tubular lumen in exchange for sodium, which is reabsorbed. The bicarbonate is transported across the basolateral membrane along with the reabsorbed sodium into the interstitial fluid, and this again constitutes a new bicarbonate being added to the blood. The figure shows the production and secretion of ammonium ion by proximal tubular cells. Glutamine is metabolized in the cell, yielding 2 ammonium ions and 2 bicarbonate ions. The ammonium ion is secreted into the lumen by a sodium ammonium ion exchanger. Note that for each glutamine molecule metabolized, 2 ammonium ions are produced and secreted in urine, and 2 new bicarbonate ions are returned to the blood. In the collecting tubules and ducts, the addition of ammonium ions to the tubular fluids occurs through a different mechanism, which is like this, that the hydrogen ion is secreted by the tubular membrane into the lumen, where it combines with ammonia to form ammonium ion, which is then excreted. You can see in this figure, which shows that the buffering of hydrogen ions secretion by ammonia in the collecting tubules. Ammonia diffuses into the tubular lumen, and where it reacts with the secreted hydrogen ions to form ammonium ions, which is then excreted in the urine. Note, for each ammonium ion excreted, a new bicarbonate ion is formed in the tubular cells and return to the peritubular blood. The luminal membrane here, which is in the collecting ducts, is permeable to ammonia but not to ammonium ion. Therefore, once the hydrogen ion has reacted with the ammonia to form ammonium ions, the ammonium ion is trapped in the tubular lumen and eliminated in the urine, and therefore for each ammonium ion secreted, a new bicarbonate is generated and added to the blood. Ammonia buffer system has a great importance in chronic acidosis. In chronic acidosis, increased extracellular fluid hydrogen ion concentration stimulates a renal glutamine metabolism and therefore increases the formation of ammonium ions and new bicarbonate ions to be used in hydrogen ion buffering. Therefore, in chronic acidosis, remember that the dominant mechanism by which acid is eliminated is by excretion of ammonium ions.